Hi there, my name is Sergio Santamaria. I'm a research scientist in the Space Biosciences Division at NASA Ames in California and a research professor at the University of New Mexico. The title of my presentation today is 2020 to 2030 New Technologies for Microbial Research in Deep Space. We will talk particularly about the research that my group was involved from 2020 to 2030 as part of the Artemis program, and we'll also talk a little about of what's coming next. Some of you may still remember this graphic of NASA's journey to Mars, which shows the different steps that we use to return to the surface of the moon and preparation for going to Mars. In 2020, we were working on the commercial crew vehicles through Boeing and SpaceX, and we continue our work in preparation for the Artemis I mission that launched in 2022. Here's a little description, a graphical description of the Artemis program and what we have been doing from 2020 to 2030. As I just mentioned, we finally returned to lunar orbit in 2022 with Artemis I, which was an unmanned mission. In 2024, we finally stepped on the moon again, shortly, but we did. We still continue the experiments with the automated robotic missions, and throughout the years, together with our commercial partners, we started to work on the cislunar gateway. We are now at the stage that after multiple missions, we have been able to assemble or almost complete the gateway. We're hoping to add some of the additional components in the next couple of years with the Artemis 6 and 7 missions and start preparing for preparations to go to Mars. In preparation for the Artemis 1 mission, that used to be called EM1 for Exploration Mission 1, NASA realized that there was plenty of space, volume, mass available to add a series of secondary payloads. As you remember, the primary payload, of course, was the Orion multi-crew vehicle. That is why there was a whole of a selection process in which 13 secondary payloads, also known as CubeSats or small satellites, were chosen to fly on the Artemis 1. Many of these missions had a series of objectives, for example, to look at water deposits on the surface of the moon, or heliophysics experiments, or even propulsion or demonstration of new technologies. There was one mission in particular called BioSentinel that tested for the first time the response of a biological organism in deep space. I was fortunate enough to be the project scientist on that mission. As I just mentioned, as you can see here, this is part of the stage adapter, which is pretty much right underneath Orion. And you can see in green each of those secondary payloads or CubeSats that were sent on Artemis 1. On the left, you can see the picture to give you an idea of size compared to humans. So after the Artemis 1 mission and the launch of BioSentinel, we were able to send multiple additional missions. I'll talk about the ones that I was involved directly and some of the ones that we worked through collaborators for other biological experiments. We also had the opportunity, as part of Artemis, to send a series of experiments to the surface of the moon using the human lander system as well as the commercial lunar landers. Before I talk a little bit more about BioSentinel, let me just go a little bit back in history of biological CubeSats. As you know, there have not been really that many of them, And everything started with GeneSat-1 in 2006, uh, almost 25 years ago. The objective of GeneSat-1 was to use the bacterium Escherichia coli to study gene expression in microgravity. It's important to mention all these missions here were sent to lower orbit compared to BioSentinel, which was the first deep space biological mission. Uh, After GeneSat-1, we had PharmaSat, which sent the GSat Artemis I-VI, which was also used in BioSentinel, in this case to study the response to drug exposure in microgravity. The next mission was ORIOS, or Organism Organic Response to Orbital Stress, which was more of an astrobiological mission looking at survival and metabolic activity in Bacillus subtilis. After ORIOS, we launched SporeSat, that unfortunately had some technical issues regarding the biological experiments. However, demonstrating the use of microcentrifuges to be able to produce artificial gravity in space, which was particularly interesting and something we actually use in one of our missions later on. And finally, before BioSentinel, we had CubeSat, which was launched in 2017 from the International Space Station. It was actually the first 6U CubeSat that was launched or that was deployed from ISS. And the goal of this mission was to study the antibiotic resistance in microgravity using an uropathogenic E. coli. As you can see on the right side, there are a series of technologies that were used for these missions, and which actually we use many of these concepts and designs for the BioSentinel mission. Another very important CubeSat mission that we had to mention before BioSentinel was the Marco or Mars Cube 1 CubeSats, which were the first deep space satellites. Their goal was to tag along with the Mars InSight mission and to send information back to, to the ground, to Earth. Uh, as you can see on the top right, this is an actual image taken from one of the Marco CubeSats of Mars far away from the surface. Many of the components used in Marco were actually like the radio ended up being used, a very similar one on BioSentinel. Let's talk a little bit about BioSentinel. As I mentioned earlier, BioSentinel was the first deep space biological CubeSat after the Marco CubeSats. It was the first one to fly beyond lower orbit, and it launched, as I mentioned, on Artemis I in 2022. It actually contained an LET spectrometer, not just to measure the amount of radiation, but also to characterize the type of radiation particles that hit the biology throughout the 12-month mission. Something important to mention as well is that we had an ISS control experiment to study the effects in a low radiation environment with microgravity, and we finally were able to launch to put another replica of this payload on the surface of the moon on Artemis III. The goal, of course, was to study this particular environment not only for the different radiation levels compared to deep space or lower orbit, but also because of the partial gravity of the moon. And you can see here some of the components of BioSentinel. As you know, it actually flew Baringi, Saccharomyces cerevisiae, inside a series of fluidic cards. And the technology that we use, it was just absorbance to study both cell growth and metabolic activity throughout the 12 months of the mission. And as I mentioned earlier, it was a 6U free flyer or CubeSat. And an important component was a 4U biosensor payload, which was inside a BioSentinel that was used both on ISS and as part of Artemis III on the surface of the moon. And this same payload was actually used by other groups to apply to their different types of research. Here you can see the different experiments that we have performed throughout the Artemis program. On the top left, of course, BioSentinel, just simple optical measurements to study cell growth and metabolic activity, with the primary research objective of studying the DNA damage response to the deep space environment. Of course, we got great results. We got a great publications out of this mission. The next mission on Artemis II, we also were very lucky on sending another payload there, was to a variation of the payload, of this same payload, but with more fluidic components that allow us to do some fluidic exchanges with a special selective media to look at radiation-induced mutagenesis, meaning that the acquisition of mutations due to radiation in deep space. Then we were able to do a couple of experiments to study continuous culturing, meaning we were able to do multi-generational studies to look at the acquisition of adaptive beneficial mutations, meaning those mutations that favor better growth in microgravity. And we also were able to use those same microcentrifuges that were developed for CubeSat to be able to replicate artificially the gravity of Earth in space. We also were able to put this same guy on the surface of the moon, like I mentioned earlier on Artemis III. And the whole idea was just to do adaptive evolution studies. Our last mission was actually, we actually used technology developed for GeneSat, a little modified to look at fluorescence microscopy and cytometry. We're able to miniaturize a flow cytometer to look at the expression of particular genes of interest that were induced by both radiation and reduced gravity. That was a great series of experiments and we got nice publications out of it. So on the top right, you can see what we're actually working nowadays. We're hoping, now that many of these technologies have been miniaturized, we're hoping to apply some of these instruments for this small payload, in this case, this same 4U biosensor payload. One of those experiments we're very proud of is using this concept of dielectric spectroscopy, meaning studying the bioelectric signatures. As you know, every time there is a morphological or phenotypic change in at least in yeast and bacteria, you can see changes in the electrical signatures of these organisms, which allows you to study very transient changes, something you cannot really do via optical density or even with fluorescence microscopy. We're also starting to do some experiments of aging using single cell studies. We're using very specialized fluidics and cytometry technologies nowadays with our collaborators at Stanford University. And also we're applying some of the concepts that we learned using the MinION instruments that allow you to do genome sequencing and nowadays RNA sequencing to look at gene expression. And of course, this has taken us a long time to try to integrate not just the sequencing capability, but also cell growth and extraction of the genetic material. And we hope to apply this in the upcoming Artemis 7 and perhaps Artemis 8 missions. Well, that was my last slide. Thank you very much for your attention. Feel free to contact me if you have any questions. Enjoy the rest of the meeting.